Welcome to KJR SummaryCast. SummaryCast provides audio summaries of KJR articles created using an in-house AI tool with editorial or author review. For full details, please refer to the original article. Welcome to SummaryCast.
Today we're diving into the brain's internal plumbing, its waste clearance system. It's a topic that's gotten a lot of attention in adult neuroscience, particularly around diseases like Alzheimer's. But we're going to look at the other end of the lifespan, the developing brain.
Right. What does this system look like during infancy and childhood? And what can imaging tell us about its function? We're discussing a review paper that pulls all this together. It's titled Imaging Biomarkers of the Glymphatic System in the Developing Brain, published in a 2026 issue of the Korean Journal of Radiology.
At a high level, this system is called the glymphatic system. Think of it as the brain's cleanup crew. It facilitates the exchange of cerebral spinal fluid, or CSF, with the interstitial fluid that bathes the brain cells.
And its main job is clearing out metabolic waste and neurotoxic proteins. In adults, we know that when this system fails, bad things can happen. But what about in kids?
That's the central question. Its role during early development is, as the paper puts it, poorly understood. This review focuses on 3 non-invasive MRI-based biomarkers that act as proxies for glymphatic function in pediatric populations.
So, not measuring the flow directly, but measuring things that should reflect it. What are they?
First, the structure of the perivascular spaces, or PVS. Second, a diffusion-based metric called the DTI ALPS index. And third, the volume of the choroid plexus.
Okay, let's break those down. Let's start with perivascular spaces. What are we looking at?
PVS are these tiny fluid-filled channels that surround blood vessels penetrating the brain. They're also known as Verco-Robin spaces. On T2-weighted MRI, they show up as small, bright linear or round structures that are isointense with CSF.
They've been noted for a long time, but were often considered incidental findings unless they were massively dilated. What's changed?
The understanding that they're a key part of the glymphatic pathway. So now, quantifying them has become a major focus. The paper outlines 2 main approaches, a semi-quantitative visual grading system and a fully quantitative volumetric measurement.
Tell me about the visual grading. How does that work in practice?
It's a 5-point scale, from grade 0 for no visible PVS up to grade 4 for more than 40. Raters count the number of PVS in a single axial slice where they are most prominent, typically in 2 main regions, the basal ganglia and the white matter.
That sounds subjective. Is it standardized?
There are efforts to standardize it. For example, when assessing the basal ganglia, the protocol excludes any PVS seen at or below the level of the anterior commissure. The reasoning is that almost everyone has visible PVS there, so including them reduces the ability to detect clinically meaningful differences.
Okay, so it's a quick and dirty assessment of PVS burden. What are the limitations?
Well, rater dependence is the big one. It's also prone to ceiling and floor effects. For instance, in neonates, about 93% have a white matter PVS grade of 0. That doesn't give you a lot of room to see changes.
Right, so that leads to the second method, quantitative volumetry.
Exactly. This involves segmenting the PVS on 3D T2-weighted images to calculate their total volume. It's usually done with computational methods that enhance tubular structures, like a Frangi filter, followed by manual cleanup.
That sounds more precise, but also more challenging, especially in infants.
It is. Infant brains are smaller, the images often have a lower signal-to-noise ratio, and the tissue contrast between gray and white matter is less pronounced. All of that makes automated segmentation tricky.
So what's the solution? Just more manual work?
Not entirely. The paper mentions that specialized processing pipelines like Infant Free Surfer and BabySeg have been developed to improve regional brain segmentation, which is a necessary first step for accurate PVS extraction.
The review also mentions ultrasound for PVS assessment. That seems unique to the neonatal setting.
It is. A study found that PVS in parts of the basal ganglia appear as hyperechogenic structures on cranial ultrasound. The volume measured this way actually correlated well with MRI-derived measurements.
Interesting. So it's a potential bedside tool, though the paper notes it's looking at slightly different anatomical regions than the typical MRI studies. Needs more validation.
Precisely. Let's move to the second biomarker, the DTI ALPS index.
Okay, if PVS volume is about structure, I'm guessing this one is more about function.
You nailed it. The DTI ALPS index is designed to measure water diffusivity along the perivascular spaces, which is thought to reflect the efficiency of fluid movement or glymphatic clearance.
How is it calculated?
It's a ratio derived from diffusion tensor imaging, or DTI. You place regions of interest at the level of the lateral ventricle body. The index is the ratio of water diffusivity in the direction of the PVS to the diffusivity in the direction of the main white matter tracts, like the projection and association fibers.
A higher ratio would mean more water movement along the PVS relative to the surrounding tissue.
That's the idea. And computationally, it's less demanding than PVS segmentation. DTI is often part of routine pediatric neuroimaging protocols.
But it sounds highly operator-dependent. Where you place those ROIs seems critical.
It is, and that's a major limitation. The challenges are amplified in neonates because their brains are so small and the anatomical landmarks are less distinct. Researchers have to shrink the standard adult ROI size from 5 millimeters down to 3 or 4.
That introduces a lot of potential for variability.
Absolutely. Inter-observer variability is a significant concern, and the paper flags that this needs to be considered when interpreting DTI ALPS findings in kids.
Got it. And the third biomarker was choroid plexus volume. What's the connection there?
The choroid plexus, or CP, is what produces CSF. The thinking is that its volume might be a surrogate for CSF secretory capacity. Since CSF turnover is the engine driving glymphatic clearance, a healthier, larger CP might indicate a more robust system.
Okay, how is CP volume measured?
Unlike PVS, there's no standardized visual grading system. It's all about quantification from MRI, either with manual segmentation or automated deep learning models.
What's the best imaging sequence for this?
High-resolution 3D T1-weighted images are generally preferred because they provide excellent contrast between the CP tissue and the surrounding ventricular CSF. The paper notes that T2-weighted FLAIR can actually overestimate the volume.
So we have these three markers: PVS volume, DTI ALPS index, and CP volume. The really interesting part is how they change as the brain develops. What does that trajectory look like?
It's dynamic and not always intuitive. Let's look at Figure 2 from the paper, which summarizes these developmental trajectories from birth to 18 years.
Let's start with PVS volume.
In neonates, the BG-PVS fraction, that is, basal ganglia PVS volume normalized to the size of the basal ganglia, actually decreases as postmenstrual age at MRI increases. In contrast, absolute PVS volume in neonates does not appear to change significantly with age.
So the basal ganglia are growing faster than the PVS within them. And what happens after infancy?
The trend of decreasing PVS volume seems to continue. A study of individuals from age 8 into their mid-30s found a negative association between PVS volume and age. It suggests a continued reduction throughout childhood.
But there's a big gap in the data, isn't there, between the neonatal period and age 8?
A very big gap. The paper highlights this as an area needing much more research. The trajectories in Figure 2 are extrapolated across that gap, shown with dashed lines.
What about the DTI ALPS index? How does that change over time?
It goes in the opposite direction. It increases with age. Studies show it's positively correlated with postmenstrual age in neonates and continues to increase through childhood and adolescence.
Hold on, that's a red flag for me. The white matter itself is maturing during this time. Myelination is increasing, which dramatically changes DTI metrics. How do we know the change in DTI ALPS index is purely about glymphatic function and not just a reflection of underlying white matter development?
That is a fantastic point and a major confounder that the paper explicitly discusses. Changes in WM geometry, like crossing fibers and axonal organization, can affect DTI ALPS measurements. So a simple interpretation of higher is better across different age groups is risky. It's a critical caveat.
Okay, and the third trajectory, choroid plexus volume?
The data there is a bit mixed. CT-based studies suggest rapid growth in the first year of life, then a plateau.
But a more recent large MRI-based study found that CP volume is actually pretty stable for the first 2 years of life, and then increases progressively and linearly throughout childhood and adolescence.
So, a complex and somewhat incomplete picture of normal development. How do these biomarkers behave in pediatric disorders? Let's start with preterm birth.
Preterm birth is a major risk factor for neurodevelopmental issues, and it appears to be associated with alterations in the glymphatic system. But the findings, especially for PVS, are not straightforward.
What does the evidence show?
It's conflicting. One study cited in the review reported that preterm neonates had lower basal ganglia PVS visual grading scores and lower PVS volumes compared to term neonates.
Okay, so smaller PVS in preemies.
Well, not so fast. A more recent study found something different. It reported no difference in the basal ganglia PVS counts, but actually found higher white matter PVS counts in the preterm group.
That's the opposite. So the relationship between prematurity and PVS is still an open question. That's a crucial point of nuance.
Exactly. For the other markers, the picture is a bit clearer. The DTI-ALPS index was found to be lower in preterm than in term neonates, and CP volume was smaller, though this difference became non-significant after adjusting for potential confounders like postmenstrual age.
So the association with CP volume might just reflect developmental maturity rather than prematurity itself.
That seems to be the interpretation. The next condition discussed is neonatal hypoxic ischemic injury, or HII.
A major cause of acute brain injury. What do the biomarkers show here?
One study found that neonates with HII had significantly smaller basal ganglia PVS volumes and fractions compared to healthy controls.
Smaller PVS again. What about the DTI-ALPS index?
Again, the evidence is mixed. The same study that found smaller PVS reported no significant difference in the DTI-ALPS index. However, another study did report a lower DTI-ALPS index in neonates with HII.
So, more conflicting results. It feels like this field is still in its very early stages.
That's a fair assessment. There's also very little data on choroid plexus volume in HII, which the authors flag as an area for future research, given the CP's vulnerability to this type of injury.
Let's turn to neurodevelopmental disorders, like autism spectrum disorder.
Here, the findings for PVS are different. Instead of being smaller, studies have reported a higher prevalence of enlarged PVS in infants later diagnosed with ASD. Another pilot study linked increased BG-PVS volume in neonates with poor cognitive and motor outcomes at 24 months.
That's a complete flip. Smaller PVS in HII, larger in ASD. That suggests the mechanism of glymphatic disruption could be very different across conditions.
It certainly does. And consistent with the idea of dysfunction, the DTI-ALPS index was found to be lower in children with ASD or ADHD compared to healthy controls.
And CP volume?
The paper notes that one study found increased CP volume in adults with ASD, but data in pediatric patients is still limited. The review provides a great summary in Table 2, showing these patterns across different disorders like epilepsy and headache as well, generally pointing towards increased PVS and decreased DTI-ALPS in pathological states.
This all brings us to the challenges. It's one thing to find these associations, but another to use them clinically. What are the major hurdles?
The paper lays out several. First, there are the technical challenges of imaging children. Patient motion is a huge problem, degrading image quality and complicating segmentation of these very small structures.
Sedation can reduce motion, but that comes with its own risks and concerns.
Exactly. And beyond the technical aspects, there's a major scientific limitation, a lack of pathological validation in children. In adults, post-mortem studies have provided histological confirmation for some of these imaging findings. That's largely absent in pediatric research.
Which means the interpretation is still on shaky ground. For example, enlarged PVS. We're assuming it means impaired CSF-ISF exchange, a sort of glymphatic traffic jam. But couldn't it also just be a sign of brain atrophy?
That's a critical point. The space could be larger simply because the surrounding brain tissue has shrunk. Differentiating between those two possibilities from imaging alone is a non-trivial problem. Neuroinflammation is another potential cause of enlarged PVS.
And the DTI ALPS index has its own interpretation issues, as we discussed. It's highly sensitive to imaging acquisition parameters, and more importantly, to the normal developmental changes in white matter.
That's right. Cross-age comparisons have to be done with extreme caution. You can't just apply adult thresholds or assumptions to pediatric data.
So if we pull all this together, what's the bottom line? Where does this leave us?
The review concludes that these non-invasive MRI biomarkers are promising. They show dynamic changes during normal development and appear to be altered in a range of significant pediatric conditions, from preterm birth to neurodevelopmental disorders. They have potential for early identification of at-risk children.
But, and it's a big but, the field is held back by fragmented and limited normative data. We have some snapshots from the neonatal period and from older children, but that huge gap in between during early childhood is mostly a black box.
Precisely. To move this from a research tool to a clinical one, the authors stress the need for a few key things. First, standardized imaging protocols to make data comparable across centers.
Second, large longitudinal studies to establish age-specific reference ranges. We need to know what's normal before we can confidently say what's abnormal.
And third, studies that can clarify the prognostic significance of these biomarkers. Does an abnormal PVS volume or DTI ALPS index in infancy actually predict long-term neurological outcomes?
The pilot study on PVS and 24-month outcomes suggests it might, but that needs to be validated in much larger cohorts.
Exactly. So, in summary, the tools to image the developing brain's glymphatic system are here, and they're revealing intriguing patterns. But the foundational work of building robust normative datasets and validating their clinical utility is just beginning.
A field with a lot of potential, but also a lot of work still to be done. That concludes this episode of SummaryCast.